Hello, everyone. As you're walking through the virtual portal to our lecture hall, I'll stall for a couple minutes. I wish I could see you. A nice group of people is walking through right now. I'll watch the numbers as they rise, and we'll get started as soon as the seats seem full. And we're at 73 already, which is great, still going up. It is kind of like watching the election results here. Getting there. Okay, I think it's about time to get started. Good afternoon, everyone. My name is Rick Page. I'm Dean of UVM's Larner College of Medicine, and I'd like to welcome you to our 5th annual State of Research at the Larner College of Medicine talk as part of our celebration of research excellence. This is an event that we've always live streamed out beyond our campus. This year, we've transitioned to Zoom, and we're all part of the live stream. I wish we could gather together in person, but I'm grateful we have this technology that allows us to connect in this way. The Dean's Excellence in Research Celebration is a 2-day event that grew out of our strategic commitment to recognize accomplishments in research at our college and our academic health system. Yesterday, many of you had the opportunity to hear our graduate students present some truly outstanding research. Later in the day, we recognized UVM Medical Alumni Association Distinguished Graduate Alumni Award recipient for 2020, Dr. Marilyn Cipolla. Dr. Cipolla provided a view of her science, her career journey in a talk entitled Lessons I've Learned, How Mentoring and My Dogs Helped Shape My Career. That's already online. If you missed it, please check out that wonderful talk. Today, we continue the celebration, recognizing a number of individuals with our annual awards. And after that, we're going to hear our keynote speaker, our own university distinguished professor, Dr. Mark Nelson. First, I'd like to offer a special welcome to UVM Provost and Senior Vice President, Dr. Patricia Prelock. Provost Prelock is a true partner to our college and our research success. It means so much to us to have her be part of today's celebration. I'm very pleased now to introduce Dr. Provost Patty Prelock. Dr. Prelock.
Thank you so much, Rick. I'm so delighted to join you this afternoon to celebrate the many research accomplishments of the distinguished faculty of the Larner College of Medicine. Thanks in large part to the significant contributions of the Larner College, the University of Vermont has really built distinctive research strengths that align with the urgent and interdependent need to support both the health of our environment and our society. Your solutions, your discoveries, your innovations, your inventions improve the quality of life for individuals and communities across the state, our nation, and our globe. And there's no doubt that the success of the Larner College of Medicine is then UVM's success. Your research accomplishments also leverage the university's corporate, philanthropic, and federal support. You help multiply our impact as a land-grant institution. You've helped enhance our reputation, and you provide enriching opportunities for faculty and students across campus. I've actually seen the commitment to student and faculty research and mentorship firsthand in the number of undergraduate and graduate students you support throughout the semester, year-long, and in summer research experiences. And your willingness when I was actually the Dean of the College of Nursing and Health Sciences to step up and mentor and support the junior faculty that I hired, helping them to really launch successful research careers. For that, I'm forever indebted to my Larner College of Medicine colleagues. And speaking of your success, it's my great pleasure to acknowledge the Larner College's contribution to the university's recent external funding accomplishments. Learning that we had achieved our most successful research funding year ever was one of the true highlights of a very challenging 2020. And you can be sure I tweet it out and I talk about it a lot. You are key to our research success. I could also not be more proud of the contributions the Larner College physicians and alums who are on the front line of treating patients and protecting public health, and the myriad contributions of Larner researchers who are doing lots of things like uncovering information about COVID-19 immune response, developing new testing methods, using computer modeling to help battle the virus, helping us understand how to manage our natural urge to socialize, highlighting the impact of COVID-19 on tobacco use, not to mention inventing an emergency ventilator. And that's just some of the incredible work you have been involved in. Your quick pivot and your willingness to bring your knowledge, your skills, and your keen ability to navigate, learn, and excel through the pandemic, for me, has been truly awe-inspiring and a timely and true embodiment of our land-grant mission. So thank you so much for all that you do, for having me here today to celebrate one of my favorite topics, which is research. I'm so honored to be a part of the celebration, and I'm very eager to learn more about the important work that's happening at the Larner College of Medicine. Thank you, Dean Page.
Thank you so much, Provost Prelock. Now, growing and maintaining a successful research enterprise can only come about with strong cross-campus collaboration, as you just heard. We're so fortunate to have connections with the university's Office of the Vice President for Research. This year, UVM welcomed a new Vice President for Research, Dr. Kirk Dombrowski. In a short time, Dr. Dombrowski has demonstrated a very quick uptake in terms of learning about our college, our university, and the Burlington environment. And just in a very short time, despite the challenges of the pandemic, he's demonstrated himself to be a dynamic leader and a terrific partner to the Larner College of Medicine. I'm so pleased he's able to join us here today. Please join me in welcoming Dr. Kirk Dombrowski.
Thank you very much, Dean Page. And I don't want to steal the thunder. I know Gordon is going to talk about all the amazing accomplishments from the college this year. We've been working on and watching them all grow together. I wanted to just give you a little bit of a congratulations and talk to you about how much we're really excited about what's happening there. We've had a tremendous research growth in terms of total awards and in the other metrics, things like business startups, total proposals. If you were to sum it all up, our external funding for the university is up more than 25% year on year. Proposal dollars are up more than 80%. The Office of Research this year processed 1,302 grant applications. We managed 680 new external awards, 2,698 active IRB protocols, 250 subawards, 900 financial reports, 152 animal safety protocols, 45 invention disclosures, 43 technology licenses, and 33 provisional and non-provisional patent applications. It's been quite a year. Our Office of Instrumentation reported this year that they performed 3,608 instrument repairs and research equipment procedures. So as a partner for the College of Medicine, we're there with you working every day. We've all faced unprecedented challenges this year in areas like animal care and protecting critical research assets, and we've come through with tremendous success. Following the shutdown in late March, we brought our entire research capacity back online in a matter of months. I think Gordon and I talked probably about 3 times a day for the first 2 months I was here. And I do have to say especially a thank you to Gordon. Starting when I did and in that situation, I can't imagine a better partner, mentor, and colleague than Gordon was that time. We're just tremendously fortunate to have him here. Now I lost my note. So we have individual safety plans for every lab that was meticulously reviewed by both your dean's office and by my staff. And we've met every compliance and research protection deadline this year on time, which was amazing. Finally, I'm really excited to announce our pursuit of Carnegie R1 status as part of the President Garamella's Amplifying Our Impact plan. We've developed a detailed plan for that, and it's been widely vetted, and I look forward to presenting that plan for your consideration in a couple of meetings we have coming up in the next couple of months, Rick, right? And so with all that in mind, and without trying to steal any of Gordon's thunder as he talks about all the amazing things that have happened this year, I just want to say congratulations and what a pleasure it is to be here and to be working with you all. Thanks.
Thank you, Dr. Dombrowski, and it's a pleasure to be working with you. We'll now hear an update on the state of our research mission, and then spotlight research excellence across the board of the Larner College of Medicine and the UVM Health Network Medical Group. We'll acknowledge both the scientific inquiry taking place right now throughout our institution and its partners, and the important mentorship and training that helps create the research community of the future. A successful, vibrant research community doesn't happen by accident, but through careful planning and hard work. Today, we have the opportunity to celebrate that vision and the dedication of the members of our research community. But first, on to the Where Are We Now part of the program. And for that, I'd like to welcome to the spotlight Dr. Gordon Jensen, our Senior Associate Dean for Research and Director of Research for the University of Vermont Health Network. He's going to share a deeper view of the current state of research at our institution. After that, he and Dean Berger will present today's awards. Please join me in welcoming the person who's really responsible for putting together this event, Dean Gordon Jensen.
Well, thank you, Provost Prelock, Vice President for Research, Kirk Dombrowski, and Dean Page, for your warm comments and support. I want to start by acknowledging that many of our medical center colleagues cannot attend today because of the computer attack that we suffered. And I'm very sad that they're not able to participate in this event today because they can't connect. But we will disseminate my presentation both on our website, and we'll also send it as a PDF to everyone. So what we've already heard from is Dean Page, Provost Prelock, and VP for Research, Kirk Dombrowski. Next, I'm going to give a nice overview of the state of research at the college, followed by the research celebrations, and then Mark Nelson's keynote address. So I thought I would just wade right into where we're at. And to me, that meant I really had to start with the pandemic. And if we go back and look at the timeline of what we experienced here, as Kirk said, we really embarked on a very immediate closure, March 25th. What I quickly learned is that was hard. But what's a lot tougher is a well-planned phased reopening. Closing is one thing, reopening is another. But we really were quite successful. So March 25th, we closed down. March 26th through the start of June, really focused on essential research personnel and activities only. A lot of folks working remotely. First couple of weeks of June, we added additional research personnel and activities. And really from June 15th to today, we are effectively open for really almost all research activities, both in our labs as well as in the community and clinical. And we've been carefully following university and Vermont health guidance. The phased reopening for clinical research was pretty similar, but an even more protracted timeline, as you might expect. So really, since the middle of June, we have been among the most fully functioning research institutions in the United States. And we really had quite a dramatic response to all this by our investigators. We had a wonderful COVID-19 research slam that Beth Kirkpatrick directed for us a couple months back now. We had tremendous attendance, short bulleted presentations and some Q&A, and this sparked a number of exciting collaborations. To date, our researchers have initiated 57 related proposals and projects, including 16 NIH proposals, 13 administrative supplements, 1 competitive revision, and a handful of completely new proposals. I know Provost Preluck did this a little bit. I want to highlight just a few of the standouts in this process, and I apologize to those that I've left out. But first, I want to mention Drs. Emily Bruce and Jason Button had a tremendous paper in PLOS One biology very early in the pandemic that showed that a RNA extraction step in the PCR testing for the virus could actually be skipped. Another paper in collaboration with Dr. Majumdar just came out in Cell, looking at how the virus disrupts splicing, translation, and protein trafficking. You heard about the Vermontilator developed by Dr. Jason Bates, an innovative ventilator based upon airway pressure release ventilation. This has attracted incredible investor interest and was even featured in the New Yorker magazine. I wanted to mention Russ Tracy, who is actually running the entire central lab for NIH Active for antithrombotic program investigation with 3 trials of interventions. Mary Cushman is the co-PI and central lab director for a multi-site rapid COVID anticoagulation trial that's funded out of the University of Toronto. Neil Zakai is partnering on a large CDC study with 10 hospital systems, 23 hospitals, looking at bleeding and thrombotic complications in hospitalized COVID-19 patients and whether antithrombotic therapy is helpful. Sean Deal's NIH-supported work has led to some new insights in regard to antibody responses during infection. This was recently published in Clinical and Translational Immunology. And Dr. Elena Vandenbroek-Altenberg and colleagues contributed a paper on risk factors in COVID-19 community exposure and mask wearing. This is a fun paper to read. It was done on a local cohort here, so you have a chance. Take a look at that. So those are just a few of many. The next thing that I wanted to highlight is the vaccine trial with AstraZeneca that Dr. Beth Kirkpatrick and colleagues have initiated. This was just announced October 27th. And they're recruiting volunteers right now. Couldn't be more timely. I know that they have additional vaccine studies in the hopper. Turning to more recent pandemic data, this is from Vermont Health and shows the daily prevalence of infection in the state of Vermont. And I think everyone's aware that we've had a more recent surge in infections. And of course, this is beginning to roost right here in our county with some 35 cases today, half dozen student cases yesterday. I added this slide late last night in
Dr.
response.
Dean Jensen,
Yeah.
we're having a bit of a technical difficulty in sharing slides.
Well, let me try it again. I wonder why it went out.
And you might try to reshare slides from your computer.
Yeah, hang on.
We were sharing for you, but that's gone out.
All right, hold on a minute. I don't know why it did that. Okay. How's that? Look any better?
That looks good.
Okay. My apologies. It didn't show a problem here, but interesting. Let's try again and let me know. Just real quick, the slides that you might have missed here, the last couple. We talked about the vaccine trial. We talked about some of our research breakthroughs and the very latest pandemic statistics. And this is a slide that I added just last night in response to what's happening on campus. So going forward, and you'll see a bunch more messaging about this, but we really are going to emphasize social distancing, masking, and hygiene. Everyone's been doing a fantastic job in that regard. Continue to really limit travel. Quarantine is indicated. Our college facilities will remain locked down. Testing is readily available nearby on campus. We have an OVPR leadership committee that meets weekly to review the campus, local, and regional caseloads and percentage of positive tests. From the VP for Research's office, we will release instructions as appropriate, following Vermont Health and the governor's office guidance. We have a new 5-phase template of opening and closing that is very similar to what we did back in March, April, May, and June, and very similar approach, but we will definitely keep everyone fully apprised. With the upcoming break, with Thanksgiving all the way through to later in January, detailed instructions will be forthcoming. Some are already out. We hope to continue to be fully engaged in our research activities, but please be prepared for any possible ramp down depending on the progression of the pandemic. For the moment, we are most certainly planning on still allowing selected undergraduates, our grad students, postdocs, faculty, and staff to continue to work in our laboratories, but we will keep you closely informed. So jumping from the pandemic, let's go to the Firestone Medical Research Building. First, just a key events timeline. Our building was approved by the Board of Trustees back in January. Original construction start was to be in April. That was delayed to October because of the pandemic. And project completion at this point is targeted for winter of 2023. Current status is that the given parking lot has been established as a construction site. No one could have missed that. They are busy working on utility work, focused on electrical and site drainage work, and construction of a new Jeffords East parking lot is proceeding and should be ready by early December. The picture there is a screen grab from our live construction webcam, perhaps not as exciting as some webcams, but you can certainly check that out anytime you're interested in seeing what's happening. And we also have a website that you can access for additional information, questions, and concerns. And Eric Gagnon has just been doing a tremendous job messaging on this project. Don't hesitate to reach out to us if you have questions. I want to touch on the new Center for Biomedical Shared Resources. The cores that are participating in this are integrative genomics, the Vermont Biomedical Research Network, flow cytometry and cell sorting, microscopy, imaging, and mass spectrometry. And then bioinformatics and biomedical statistics will be in very close proximity. Our goal is to promote long-term sustainability through efficiencies of scale, improved access, sound fiscal management, cross-training of personnel, and sharing of resources. And this proposed center will encompass the entire first floor of the new Firestone Medical Research Building. Supporting this is a new $5.4 million NIH CO6 biomedical facilities grants. And the folks that were just instrumental in helping me secure this grant are Eric Gagnon, Geraldine Haraldson, Doug Todges, Julie Dragon, Roxana Del Rio Guerrera, Rex Forehand, Dwight Matthews, and Peter Callis. A super team and a wonderful reward for everyone's hard work. Touching on our new hires for the fiscal year shown here, and you can see that we do have a few for FY20. The vast majority of those predate the pandemic. We've managed to pry open a few new key recruitments. though by and large, we remain really mostly limited in terms of starting new searches for hires at this time. Peer-reviewed publications shown here, and as you can see, we've continued to hover right around 1,000 each year. What's reassuring, this most recent one, the 981, these are actually done by Steve Goldman using academic analytics for the very first time. All the prior years you see there, all the department heads hated me because it really came down to hard labor at the department level to try to identify our papers. And we finally got it done digitally in a very good way. And actually I was quite reassured to see the numbers are in the same ballpark. In terms of major grant awards for FY 2020, they're shown here. I'll let you read them yourself, but really these are remarkable accomplishments by Stacy Sigmund, Judy Shaw, Steve Higgins, Stacy Dublier, Russ Tracy, Steve Higgins again, Beth Kirkpatrick, Thompson Patico, Davi Bach, and Gary Stein. And as people have already highlighted, this was an absolute record year for the College of Medicine at $105 million. It was 58% of the extramural funding for the entire campus. This slide just summarizes our COBRA grants and our CTR grants. So our active current COBRAs include the Vermont Center on Behavior and Health under Steve Higgins, the Translational Global Infectious Disease and Research, COBRA or TIGR, directed by Beth Kirkpatrick, our brand new Vermont Center for Cardiovascular and Brain Health. I'll talk about it in a moment. And the CTR overseen at the Vermont end by Gary Stein. So the wonderful new Center for Cardiovascular and Brain Health, co-directed by Mary Cushman and Mark Nelson and funded for approximately $12 million over 5 years, seeks to better understand and treat and prevent disorders like cardiovascular disease and dementia. Neil Zakai and Peter Durda and Todd Clayson direct cores in the new COBRA, and the junior faculty mentees include Catherine Chung, Masayo Koide, and Denise Peters. So we're all really very excited about this new COBRA, a wonderful accomplishment. A couple of quick updates about the CTR. As I mentioned, Gary Stein is the co-PI for University of Vermont. One exciting new initiative, cross-campus initiative, is a collaborative gun catalyst award that we funded targeting the rural environment and health. This was a collaboration of the CTR, LCOM, Maid Medical Center, and the Good Institute. It's a $100,000 pilot for 2 years. It was awarded to Dr. Meredith Niles in Nutrition Food Science, and is looking at the impact of the COVID-19 pandemic on food security. Other exciting new projects are going to be testing a peer-based strategy to overcome HPV vaccination inequities in rural northern New England. And we're also looking to develop a rural health communication network in collaboration with Vermont Health to disseminate evidence-based pandemic and health information. So these are examples of exciting cross-northern New England projects. I also want to give a quick update on the Vermont Center on Rural Addiction, directed by Stacy Sigmund. The first year of funding, they completed baseline assessments of provider needs in Vermont. They set up a best practices provider scholarship program. They provided training and technical assistance to more than 500 participants from 42 states, did a bunch of direct clinical peer mentoring, and they distributed Narcan, fentanyl tests, masks, gloves, digital thermometers. This initiative off to a tremendous start. As I said, led by Dr. Sigmund, with a whole group of powerful collaborators. A quick update on the Center for Health Services Research, directed by Adam Atherley. They have a new affiliate membership program. They had a tremendous health services research slam a few months back that quite a number of us attended, again, promoting collaboration. Adam mentioned he'd love folks to reach out to participate. They plan to do more. They've got a host of active research projects, all the way from very narrow clinical questions to now state and region-wide analyses with multiple projects across the medical college. They've been looking at COVID capacity planning for the health system and state. They did a really fun analysis of deferred care during the pandemic. People who needed urgent cardiac care, GI care, what have you, weren't coming to the hospital, to the emergency room. They've been growing their grant funding. They have data projects promoting use of vCures, and continue to work to develop supporting infrastructure for an electronic record repository. So we've been talking a bit about some of the programs and investments, health services, the new Firestone building, our research cores. I want to touch a little bit on research support, on support for PIs of major grants, our training grants, some pilot funding, the network, and medical student research. So we have a host of cores and services. This is the website you can go to learn more about them. As you folks are aware, I've been very focused on trying to really emphasize and support our shared resources. I review every single proposal that goes through the college. I can tell you that our success in extramural funding is often very dependent on these resources. In terms of clinical research support, this is some of the data that Kim Lubers, our Assistant Dean for Clinical Research and Director of OCTR and CRC, provides to us every year. For FY20, they worked on 187 different clinical research agreements, supported 183 projects in the Office of Clinical Trials, generated a net profit of just over $90,000, half of which went back to the departments, and they conducted 9 quality assurance reviews. In the CRC, they deliver a host of resources and services listed here. And even with the pandemic, they supported 53 protocols and over 2,100 outpatient visits. So really very busy. And I would say with the pandemic and supporting the new vaccine trial, busier than ever. I think folks are aware that we have research navigators in place now that you can reach out to for assistance with identifying resources, collaborators, training, funding opportunities. The graph on the left shows the increased activities of the navigators, and the graph on the right shows the rationale why they were being consulted. Number one was education and training, but they're also being contacted to access Red Cap to support grant submissions, identify collaborators, and so on. So I would really encourage you to reach out to these folks. One of the really important new things that we're offering through Kim's team is the required course in fundamentals of clinical research that is now required for all clinical research coordinators. It was launched in February of this year. Then the pandemic came. Nonetheless, since this is, they can do this virtually, they've conducted 6 full offerings, and 65 individuals have already participated. It includes part of the city program, but it also there is a classroom competency component and assessment. And for the spring of next year, we're looking to develop a training program for clinical investigators as well. And if you need to reach the navigators or Kim Lubers, phone number, email, and a website are shown here. And as I said previously, we'll disseminate my slides to everyone, so you'll be able to find this information. In biomedical statistics, which Peter Callis directs for us, we are continuing our program to provide up to 5 hours of consultation for LCOM faculty members and trainees. You can reach out to Laura Clayton at the email address shown here. Kirk Dombrowski and I are working on a broader voucher approach so that we can provide a similar free consultation approach for the entire UVM campus. And as we develop the Center for Biomedical Shared Resources, we plan to integrate the biomedical statistics core right into that. In terms of salary support for PIs of major institutional grant opportunities, this program is thriving. Provides, we budgeted about $100,000 annually to provide 20-40% effort of PI salary at the NIH cap for up to 3 months. We typically have had several faculty members a year take advantage of this. The instructions are shown here, and you can reach out to me to submit an application. And this support has already been associated with a number of successful COBRE and training grant applications. We're also supporting salary for the PIs of actively funded training grants at 5% effort of funded T32 training grants at the NIH cap for up to 5 years corresponding to the funding. If there's more, if they're co-PIs, then it'll be split. The mechanism to request support is described here. And we've had a number of training grant PIs who are already receiving some support from us by this mechanism. So don't hesitate to reach out to me. Pilot research funds, we aim to continue our internal pilot and bridge support programs. I do want to acknowledge the assistance of Matt Wargo, who's the chair, and other members of the research committee who assist with reviewing these. In the context of the CBSR grant, we're going to try to continue the targeted pilot research program that will target new methodologies in basic research in conjunction with a research core. And these proposals require an investigator and a core director working together. So wrapping up now, there are also a host of UVM Health Network research opportunities that we're interested in. We're holding a retreat November 20th virtually, so reach out to me if you might have interest in participating. In this retreat, we're really trying to focus and identify key leaders and stakeholders throughout the network. We're going to talk about ongoing network research projects. We're going to talk about Trinetics, which is a big pharma electronic record database that we're bringing online, as well as our plans for a repository. And the overriding priority remains to enhance support and capabilities for clinical trials across the network. We also just this year implemented a medical student research fellowship that we have internally funded as a gap year for years 3 and 4. We undergo a competitive review process, and the awardees for 2020 were Jennifer Holland, who's working with Dr. Valley Harder, and Jordan Munger, who's working with Dr. Neil Sakai. Our strategic planning now is focused on Vision 2025. There are research objectives in this strategic plan, including improving infrastructure, sustaining our shared resources, enhancing data processing and storage, building on our current strengths and future opportunities, supporting innovation and entrepreneurship, strengthening mentorship and career development, maximizing faculty recruitment and retention, and I would add staff to that as well, and diversifying our funding portfolio. Some specific opportunities for our biomedical research include growing regional and national partnerships, promoting strong interdisciplinary teams to take on complex medical and public health concerns, targeting rural health disparities in New England as listed there, supporting research and training targeted COVID-19 and future pandemics, identify and support evolving research methodologies and technologies, and continuing on our work to develop an electronic health record repository. So that was a hasty overview, obviously with a bugaboo in terms of screen sharing in the middle there that was out of our control. Again, we'll disseminate all my slides to everyone. Please don't hesitate to reach out with any questions or feedback. My email address is there. That is Tigger, the wonderful mountain hiking dog who weighs all 20 pounds and stands barely 20 inches high and goes everywhere. I wanted to wrap up the State of the College research address by really affirming that these are very challenging times. And I wrote you this little note that basically says, "Dear colleagues, please accept my sincere thanks for all of your contributions to our research enterprise and to the health and safety of our community. All the best, Gordon." And indeed, you have my sincere acknowledgement and thank you. So take a breath and we'll turn to the Dean's Excellence in Research Awards. I want to start by recognizing Marilyn Cipolla, as Dean Page also did, who received our Distinguished Medical Alumni Award that was presented yesterday. And her presentation was just a hoot, really it was perfect for the intended audience, and was entitled "Lessons I've Learned How Mentoring and My Dogs Helped Shape My Career." And of course, I told Marilyn I would include a dog picture as well. And so we're going to zip through these. I'm going to turn, the first one will be the Graduate Student Research Showcase Awards, which I'll turn over to Dean Berger. And then I'll rip through the other trainee awards, the Network Medical Group Awards, the LCOM staff and LCOM faculty awards. Now, please note, we're not obviously going to be personally handing out plaques or certificates. These will be coming to the award recipients via the inter-office mail. So up to you now, Dr. Dean Berger.
Thank you, Dean Jensen. I first want to congratulate all of our participants on a truly outstanding slate of presentations from top to bottom yesterday. It was truly enjoyable to be part of that. And the judges found their job to be extremely difficult in picking our winners, which speaks very much to the quality of our students and the future of scientific research that our participants will lead in the not too distant future. So first, in the junior division, our top finisher was Melissa Boucher, followed closely by Jeffrey Braybeck. And then in the senior division, the results were just as close, with Alex Thompson in first place, followed closely by Leslie Saponiak in second place. So again, congratulations to you all. I'm excited to see the contributions you will all continue to make as you progress in your careers and become the next generation of scientific excellence.
There we go. Am I unmuted now? Okay, so we're going to jump to the additional awards and the other trainee awards. And the first ones are the trainee awards for outstanding research publication and for a graduate student. And Catherine Becca, mentored by Margaret Vizard, receives this award. The citation is shown below, focused on functional effects of blocking BEGF, VEGFR2 signaling in the rat urinary bladder in acute and chronic CYP-induced cystitis. I can't hand you an award, but I would if I could, so congratulations, Catherine. Next award is for the medical student outstanding research publication. This goes to Christina Evans, mentored by Dr. Mary Cushman. The paper by Evans and colleagues entitled "Lifestyle Moderates Genetic Risk of Venous Thromboembolism, the Atherosclerotic Risk in Communities Study," with the citation as noted. Again, congratulations, Dr. Evans. These really go much quicker if we're not having to pass them out individually and have people come up to the podium. Feels funny. The next one is for the outstanding research publication by a postdoctoral fellow. This award goes to Vitor Mori, Ph.D., mentored by Dr. Jason Bates. The winning citation entitled "Cisplatin Pharmacodynamics Following Endobronchial Ultrasound-Guided Transbronchial Needle Injection into Lung Tumors and Scientific Reports." Congratulations to Dr. Mori. The next of the outstanding research publication awards for a resident goes to Dr. Stas Amato, mentored by Dr. Gary Ann. This paper by Dr. Amato and colleagues entitled Geographic Coverage and Verification of Trauma Centers in a Rural State, highlighting the utility of location allocation for trauma system planning, in press in the Journal of American College of Surgeons. Congratulations to Dr. Amato. Next, I'm going to turn to the UVM Health Network Medical Group research grants. There are two of these here. The first, let me say first that Claude Deschamps couldn't join us this afternoon because I know that he would have liked to have announced these awards personally, so I am standing in for him. The first of these, the PI is Dr. Scott Raymond in radiology with David Johnson, Rachel Oldinsky, and Bruno Suarez as collaborators, entitled A Transradial Specific Diagnostic Catheter for Cerebral Angiography. Congratulations to Dr. Raymond and colleagues. The second research grant award went to PI Jessica Centalo Gonzalez in surgery. Her collaborators, John Ramsey, Shahid Amin, Hiba Raymond, and mentored by Carlos Mariquin in surgery. Proposal entitled Using Circulating Tumor DNA to Predict Minimal Residual Disease, Recurrent Disease, and Clinical Outcomes in Patients with High-Risk Malignant Melanoma. Congratulations to Dr. Centolo Gonzalez and her collaborators. The next grant, also from the UVM Health Network Medical Group, is the Innovation Grant. The recipient of this grant, the PI, is Devika Singh in medicine. Her collaborators, Jennifer Monroe Zakaras, Patsy Kelso, and Jennifer Reed. The proposal entitled Sexual and Reproductive Health Needs and Experiences of Women Living with HIV/AIDS in Vermont. Congratulations again to Dr. Singh and her colleagues. The next is the UVM Health Network Medical Group Faculty Practice Junior Investigator of the Year. There's actually going to be two awards. The first to Jessica Crothers, Assistant Professor of Pathology and Laboratory Medicine. Highlights of her accomplishments include research program focused on infectious disease, the microbiome, and host immune response, and effective prevention and treatment. She was awarded a WHO grant as a co-PI for a clinical trial that assessed the safety and immunologic impact of a novel polio vaccine. She was recently selected as a junior faculty project director for the COBRE on translational global infectious disease, and she already has 17 peer-reviewed papers. Congratulations to Dr. Crothers. The second Faculty Practice Junior Researcher of the Year award goes to Catherine Chung, Assistant Professor in Medicine and Nephrology. Highlights of her accomplishments: her research is focused on cognitive impairment and renal disease with a clinical focus on older adults with advanced kidney disease. She has been awarded 3 pilot grants and a career development award from the National Palliative Care Research Center for her work in geriatrics and palliative nephrology. She was selected very recently as a junior faculty project director for the COBRE on cardiovascular and brain health, and she has 8 peer-reviewed papers. Congratulations on your award to Dr. Chung. Next, we'll move to the UVM Health Network Medical Group Faculty Practice Senior Researcher of the Year Award, which goes to Dr. Elizabeth Bonney, Professor of Obstetrics, Director of the Research Division, Gynecology and Reproductive Sciences. Highlights of her accomplishments include recipient of multiple NIH grants. She is currently the lead or co-PI in 4 independent NIH Research Project Awards. She served as acting director on the board of scientific directors for the National Institute of Child Health and Human Development. She served on the Council for the Society for Reproductive Investigation. She's an active member of the Cobre Center on Immunology and Infectious Disease. She has 39 peer-reviewed papers, and she was recently selected for the executive program in academic medicine known as ELAM. Congratulations to Dr. Bonney on this prestigious award. So now turning to the Dean's Excellence in Research Awards, we implemented new research awards this year that were targeting staff. And the first go to research laboratory staff, and there were 2 outstanding candidates recognized with this award. Cheryl Collins, research analyst in the Bud Lab, nominated by Ralph Budd. She's a co-author on 18 publications, 5 as first author, and has been with the Bud Lab for 18 years. Congratulations, Cheryl. And Terry Messier, research analyst and lab manager in the Cuniff Lab, nominated by Brian Cuniff. She's a co-author on 17 papers with 25 years of UVM service, and congratulations to Terry on that recognition. The next staff award was for clinical research coordinator. This award goes to Sarah Ardren, the clinical research coordinator for the Vermont Lung Center, the pulmonary, and that is affiliated with pulmonary disease and critical care medicine. She was nominated by Renee Stapleton. She's currently the project leader for 3 multi-site investigator-initiated NIH-funded clinical trials. Congratulations to Sarah for this recognition. I want to add that we had a tremendous number of nominations for both the Research Laboratory Staff Award and for the Clinical Research Coordinator Award, and we look forward to continuing these important recognitions. Moving to the faculty category of the Dean's Excellence in Research Awards, the first is the Dean's Research Mentorship Award. This award goes to Dan Weiss, nominated by Ann Dixon. Dan is a professor of medicine in the Department of Medicine, Division of Pulmonary Disease and Critical Care. Some of Dan's accomplishments, number one, his mentees, and we solicit input from mentees for this award, highlighted specifically his passion, dedication, support, and commitment. His past mentees include 10 junior faculty, 22 postdoctoral fellows and grad students, 9 residents, 7 summer session medical students, 46 undergraduates, and 13 high school STEM students. So really an exceptional pool of mentees. He's a past recipient of the Department of Medicine Research Mentor Award, and Dan reports 86 peer-reviewed research papers, many with mentee co-authors. So congratulations to Dr. Weiss on this important recognition. Next comes another new award for this year, which is the Dean's Clinical Trials Faculty Research Award. And this is going to Harold Dowerman, nominated by Dave Schneider. Dr. Dowerman is a professor of medicine in the Department of Medicine and Division of Cardiovascular Medicine. He has published 158 manuscripts in peer-reviewed journals. He's had leadership roles in a number of major pharmacotherapy trials, including serving as the national co-PI for the EDUCATE trial. He's been very active in the development of new approaches to the treatment of structural heart disease. He's recognized for his seminal contributions in the development of pharmacoinvasive approaches to the treatment of ST elevation myocardial infarction. So congratulations to Dr. Dowerman on our first Dean's Clinical Trial Research Award. Next award is the Dean's Rising Star New Investigator Award. This is an assistant professor level award. The recipient this year is Jessica Heath, nominated by Heather Bradeen. Dr. Heath is assistant professor of departments of pediatric and biochemistry. Her research focuses on translational and molecular biology of acute leukemia. She presented a plenary paper session at the American Society of Pediatric Hematology and Oncology in May of this year. It must have been virtually at that time. She's had research funding from multiple foundation and internal pilot grants, and she has 6 peer-reviewed papers. Congratulations to Dr. Heath on receiving the Rising Star New Investigator Award. There's only a couple more of these to go. The next is the Dean's Mid-Career Investigator Award. This is an associate professor level award. The recipient this year is Jason Botten, nominated by Ralph Budd. Dr. Botten is associate professor in the Department of Medicine, Division of Immunology. Dr. Botten's research has focused on virology, including hantavirus, dengue, Zika, and more recently, COVID-19. He's had 21 plus grants as PI or multi-PI, totaling almost $10 million, 48 peer-reviewed papers to date, is recognized globally with 9 international presentations. He's been a very active mentor with multiple undergraduates, graduate students, postdocs, and junior faculty. And most recently, he's spearheading the opening of our BSL-3 research facilities in Colchester as part of their growing initiative into COVID-19. So congratulations to Dr. Botten on this Mid-Career Investigator Award. The last of our awards is the most senior, the Dean's Research Laureate Award, that is a professor recognition. This year, this very prestigious award goes to Dr. Ann Dixon, who was nominated by Renee Stapleton. Dr. Dixon is professor of medicine in the Department of Medicine, Division of Pulmonary Disease and Critical Care. She is director of the Vermont Lung Center. Highlights for her include contributions to paradigm-shifting research on obesity and asthma. She's published 120 peer-reviewed papers in high-impact journals. She's had continuous NIH funding for 20 years. Currently, she is the PI of 4 R01s and 2 R34s. She's mentored more than 25 medical and graduate students, residents, and fellows. She's served on multiple grant review panels. A very impressive body of work with important contributions. Congratulations to Dr. Dixon on receiving our Research Laureate Award. Typically, this will include a lecture in the spring. Hopefully this next year we'll be able to do it and the pandemic will be long over. So congratulations to all of our award recipients. And again, your plaques or certificates will be in the inter-office mail. And now I'm going to turn it back to Dean Page to introduce our keynote speaker. Thank you so much.
Thank you very much, Dean Jensen. I want to congratulate the awardees, nominees, and all the Research Showcase participants. It's especially meaningful that this year we're acknowledging our staff with awards. For all our 3 missions, we cannot do what we do without the support and partnership of our staff. I want to thank Dean Jensen for an outstanding summary of our research programs, and especially for coordinating our research celebration today. So now we've come to the keynote presentation for this year's research celebration. But first, a bit of housekeeping. There's a Q&A button on your screen. Please go ahead and put in your questions anytime during the talk or afterwards. These will be moderated by Dean Jensen with our speaker before he closes today's celebration. Today's speaker is one of our own, Dr. Mark Nelson, University Distinguished Professor and Chair of the Department of Pharmacology. Dr. Nelson's election last year at the National Academy of Sciences was the most recent and most prestigious of many recognitions of the value of his contribution to the body of biomedical knowledge. I'm so glad we can get to hear from him today. A member of the UVM faculty since 1986, Dr. Nelson is internationally recognized for his research on molecular mechanisms and cellular communication involving blood flow. He's a fellow of both the American Heart Association and the Biophysical Society, and received an Outstanding Investigator Award from the NIH Heart, Lung, and Blood Institute, and an NIH Merit Award. Dr. Nelson's extensive research contributions have been recognized with more than 200 peer-reviewed publications and more than 360 invited lectureships since 2000, including the Paul M. Van Hout Lectureship in Vascular Pharmacology by the American Society for Pharmacology and Experimental Therapeutics. He's received University Scholar Honors from UVM, and in 2009, he was named University Distinguished Professor, a rare recognition that honors faculty who have brought distinction to the university over an extended period of service. I should note that he was named the LCOM Research Laureate last year, but the pandemic forced us to cancel the dinner celebration and the talk that he would have delivered in the spring. And so it's fitting that today we get to acknowledge Dr. Nelson as he presents our celebration of research excellence keynote address entitled Translating Thought into Blood Flow in the Brain. Please welcome Dr. Mark Nelson.
Well, thank you very much. Dean Page, welcome everyone. I guess it's gotten dark out already. I'm going to share my slides. Is that working? There we go. So it's exciting to be here on the screen and to present a little bit about background about me and some of our work, and to really acknowledge my dear colleagues, friends, and UVM that's made all this possible over the years. I'm going to first give a PR for our new Cobra. There's Mary, you saw this earlier, as we were, actually it was for the Firestone building. And so it's a shout out to Mary and the new Cobra, and it's going to be great for the university, and it's going to focus, a bit of a unique focus, cardiovascular and brain health, neurovascular. So a little bit of background, this was a year ago, and I was actually at a meeting in Morocco. It was a legitimate meeting, of course, on calcium signaling. So there is Dave Warshaw accepting my plaque, and those are my feet at the beach, at the pool at the same time. And then later, after the meeting was over, we went on to the Sahara Desert and had to go for 2 hours on camel to an oasis. I can tell you I was in pain the whole way. So that's what I was doing a year ago, hard to believe. So a little bit about research. I had this quote in my office: if we knew what we were doing, it wouldn't be called research, would it? That's what I love about specific aims. You write them, then you forget about them, and you do whatever you follow your nose and do what you think is important. And there's another little quote from recent Nobel laureate: lack of knowledge is a true bottleneck to clinical translation. On the right is Albert Einstein again. This is a statue at the National Academy of Sciences building, which I have not seen yet. The induction was supposed to be, the election was last year, and the induction was supposed to be last April, was postponed to next April, is going virtual again, the meeting. So right now it looks like 2022. And apparently you have to do it in person. So Let me just get my pointer here. Yeah, you're supposed to do it in person because you sign the same book that everyone has signed who's been elected since 1863. And apparently everybody leased back to 1942 to look for Albert Einstein's signature, the year he was inducted. So we're going back, just a little few things from the past. Hard to believe that's me. I was pretty young. It was the end of the 70s. This was what was going on. And you can see I wasn't really involved in the disco generation. I was too busy being in the lab, and I guess I didn't have money for haircuts either. This is a preparation I worked on. It was the muscle equivalent of the squid giant axon, and it was about, this single cell is about 1 centimeter long and 1 millimeter in diameter. So you can look at the properties of single cells by using a single cell from barnacle. And I got a prop here. I know, there we go. This is a giant barnacle from Washington State. This is what I use. This is actually one of them. And this is one of my first experiments from my first paper. My advisor was Mordecai Blaustein, and he was always really enthusiastic. And he would come in and say, "Oh, this is real science." And I said, "What was I doing before?" Okay. And Morty did his postdoc with Alan Hodgkin at Cambridge, and he worked on the squid giant axon in Cambridge. And as you all know, that was the preparation that Hodgkin-Huxley used to determine the properties of excitability and action potential in nerves. So this is really dating me. This is my second paper, also on barnacle, which was in Nature, which you don't look at the date. This is my first independent lab with my first postdoc, Jennings Worley. This is in Miami. As I wrote down here, Miami Vice was on the TV. This is the mid-'80s. This is Morty's son, Mark, who came ostensibly to help me in the lab, but I think he just went out and had fun. And his colleague, Jane Talvenheimo. Fast forwarding, this is now in Baltimore. Now I'm a professor here in Vermont, and we made the first measurements of local calcium signals in smooth muscle that turned out to be very important in controlling smooth muscle diameter and blood pressure. And this is one of the first measurements. It was John Lederer, Michael Rubard, Fernando Santana, and Peace Chang. And we ended up having it in Science magazine. By the way, if anybody knows John, John hasn't aged a bit in the last 25 years. Of course, he was about 35 in the picture too. And this is one of the great passions here. This is Dave Warshaw and myself going for, well, we're actually being photographed for this article. And the guy told us to go around and around. We did it 2 days in a row. I think we did about 2 hours of running before he got one shot. Colin Freeman's in the background here. Okay, a little bit of a talk, try to make it fun. I know it's whatever day it is. I don't know about everybody else, but I was telling Dave today, I keep looking at the election results. And it's almost like looking for your grant score on ERA comments. You push the button and you hope something has changed, and then nothing has changed. Then you say, I'm not going to do this for an hour. I'm not going to do it for an hour. Then 5 minutes later, you look. So I'm not looking right now, but you guys, I'm sure are. So one of the goals of our lab is to understand the control of blood flow in the brain and how it is degraded with disease and age, and ultimately how to correct this deficit. A few factoids: the brain represents 2% of the body mass, consumes 20% of the body energy. The neurons are just-in-time machines. They have limited energy reserve and depend on on-demand delivery of blood, which is called functional hyperemia. Function because the neurons are functioning, and hyperemia means an increase in blood flow. Dysfunction of the small blood vessels in the brain, mixed with Alzheimer's pathology, is a major cause of dementia and stroke. And the societal impact and financial impact is staggering and growing as the population ages. Here's just a simulation on the left of neural activity. So right now you may be, I don't know, using your hands to sort of text somebody. And so your motor cortex neurons light up, blood has to be delivered. So if you can imagine your brain working moment to moment, day in and day out, year in and year out, it's always redirecting blood to the active parts of the brain, your visual cortex, your motor cortex, auditory cortex. And so this has to work seamlessly for your lifetime in order for you to have a healthy brain. So the question, what goes wrong and how does this work? And the basic observation of this local delivery of blood was described in the 19th century, and one seminal publication by Roy and Sherrington in 1890. This shows Roy and Sherrington. If you can identify them, well, I'll have Gordon send you something through the mail. This shows the basic features. This is from a rat, but in this case, the whiskers are wiggled. This is the barrel cortex. The nerves respond immediately because they're having sensory information. And this is the blood flow in the same area. So blood as it goes up fast and then down as the stimulation ends. And it has to be, so this delivery of blood has to be precise and rapid. And the sort of fundamental question, how does this come about? How is it controlled? And how does it go wrong? So I'm going to give you a little bit of overview, some of our work and where we're going on that, and hopefully provide some insights. This on-demand delivery of blood, functional hyperemia, is also the basis of MRI, which really is not measuring neural activity either in animals or humans, but really measuring the increase in blood flow. With the basic assumption, I just got a new Zoom light, which is kind of feeble, sorry folks, and start. But with the basic assumption that when neural activity goes up, blood flow goes up. The problem with this assumption is there's also observations of increases of blood flow where there apparently where neural activity hasn't gone up. So anyway, and I've said this before, other talks. When I was a grad student at Washington University in neuroscience, we had seminars every Saturday morning. So you can imagine for a 22-year-old to go to a seminar at 9 a.m. on Saturday morning initially seemed like fun, but then I realized I got free donuts and free coffee. So you can imagine Saturday morning, by mid-morning, we were sweating and shaking from the coffee, the caffeine and the sugar. But in all these seminars I went to, the blood vessels in the brain were never mentioned. This gives you, this figure shows a vascular cast of a human brain, and there are about 1,000 miles of blood vessels in the human brain. And on the right here shows the surface arteries that then penetrate into the brain at right angles and forms a vast network of capillaries. Capillaries are the smallest blood vessel. They're made up of one single cell type, endothelial cells, which line all the other blood vessels as well. A capillary's width is about 1/20th the width of a human hair. And what I'm going to show you, which is we've already published a lot of this, some of this, is that capillaries are actually really the sensors of neural activity to send rapid signals, electrical signals upstream to signal blood delivery. And so, and you know, it's interesting because capillaries up to recently were really thought to be only passive conduits for blood and serve in the brain as a blood-brain barrier. But if you think about it, every capillary is within 10 microns of a neuron. So they're ideally suited not only to deliver blood, but to sense neural activity. And just a little bit of another fact is that all the endothelial cells in your blood vessels are coupled by proteins called connexins that electrically couple the blood vessels. So now view your brain as an electrical network, sending signals to direct blood where it's needed when the neurons are active. So we call this a shadow nervous system, where it works to monitor the activity of the central nervous system to redirect blood where it's needed and when it's needed. So this is the basic observation. I'll show a few things to support this, but I'm not going to, you can read about it if you want. Our data work suggests that the major communicator from neurons, perhaps through astrocytes, are extracellular potassium ions. And this makes perfect sense. Every time a neuron's active, it fires an action potential, and the repolarization of the action potential is caused by the efflux of potassium from a neuron. So the more active a neuron is, the more potassium is released. This potassium then activates an ion channel called the inward rectifier in the capillary endothelial cells, which when it turns on, makes the cells more negative, which sends a signal through, electrical signals through gap junctions upstream to the penetrating arterioles, and that electrical signal goes to the smooth muscle cells, causes them to relax, and then blood flow increases downstream. And the beauty of this system, it has all the features you need for functional hyperemia. It has speed. It can be turned on rapidly. It has a retrograde electrical signal that can travel rapidly upstream to cause dilation of upstream blood vessels to deliver blood. And it can be turned off rapidly as well. So I'll just show you a couple of features of it. When we were doing this project, we thought Fabrice in the lab, Fabrice Dabertron, shown here, nice and dapper, he developed a prep where we could take an artery from within the brain, dissect it out, cannulate it, subject it to normal blood, normal transmural pressure. It will constrict, and then he left the capillary branches attached. So now we can stimulate the capillary branches at the ends of the capillary, and then look at the upstream effect on the arterioles. By the way, Fabrice left last year. He now has a tenure-track faculty position at the University of Colorado. So this shows a measurement. Here's the capillaries. Here's the arterial. Spritz potassium over here going from 10 millimolar, and then measure the upstream dilation of the arterial. And what you can see within a second, the arterial dilates. And note the size of the arterial. It's 15 microns. As I mentioned earlier, that's 1/20th the width of a human hair. So Fabrice is able to cannulate this and then do this experiment. And this response disappears when we genetically knock out the ion channel that causes it, which is called an inward rectifier potassium channel, or if we add a blocker, which in this case barium ions, that prevents this as well. I should say, it was the last slide. I wanted to call this the preparation after Fabrice, La Fabrice, but Nature Neuroscience didn't go for it. So we have the really, so they wanted, we call the Kappa prep. So the next level looking at things is in vivo. And for these experiments, which is a living mouse, we make a cranial window, and then with 2-photon microscopy, we can look at blood flow or fluorescent signals from within the brain and in the vasculature. And the beauty of this technique is you can actually image as deep as 1,000 microns into the brain, and where the 2 photons collide, they can go at longer wavelengths, and when they collide, they double the energy, and so you can look at things that fluoresce, for example, which I'll show you later on. So this shows the preparation, and what we've done is we've injected a green dye, fitzy-dextran, into the bloodstream, and now we zoom into a capillary. These are bigger surface arteries, and what you can see, a capillary is, as I mentioned, it's about the diameter lumen of a capillary is about 4 or 5 microns. So the dye here is in the bloodstream. These black dots are actually red blood cells that don't have the dye in them. So we can then determine and they go single file through the capillary because a red blood cell is about 6 microns in diameter. It's actually wider than the lumen of a capillary. And we can then measure the number of red blood cells that move through a capillary per second, or the velocity by which they move. So with this, we can measure accurately the movement of blood through capillaries in a living brain. And I'll just show you. So our mechanism predicts that the communicator from the neurons to the blood vessels, when the neurons are active, are potassium ions. So we test this by simply putting a pipette into the brain, position it at a capillary just downstream of the arterial, and then we spritz for 200 milliseconds, a little bit of potassium, just elevate it ever so slightly, simulating neural activity onto a capillary. And what you see here is a puff of red comes out. That's the elevated potassium. And you can see the movement of the red blood cells, in this case, about doubles. This doesn't happen if this potassium channel, the inward rectifier at 2.1, is genetically ablated from the vascular endothelium, or if we block it with barium. So here's the mechanism in a nutshell. Potassium's release activates this ion channel in the capillaries, causes a negative hyperpolarization electrical signal that goes from cell to cell through gap junctions upstream rapidly to cause dilation. And this is just shown in here what are called line scans. This is spritzing on 10 millimolar potassium. And you just see the black streaks increase. This is 1 second. So they more than doubled by just 200 milliseconds of exposure to potassium. So this is sort of another cartoon of the mechanism we're proposing. Neural activity leads to elevation of potassium around the capillaries. The potassium sensed causes a retrograde electrical signal that rapidly dilates upstream blood vessels. So at the same time, and this is the advantage, I would say, of international collaboration and just sort of keeping your eyes open a bit. Anne Jutel, my collaborator in Paris, contacted me about applying to the Foundation Leduc for a network grant, a transatlantic network grant, which we got, and it ended last year. It was $6 million over 5 years. And so we have groups in the US and groups in Europe and myself. You heard from Marilyn Sapoli yesterday about Frank. He was part of the network. Martin Deschamps in Munich. And so we started this collaboration. And meanwhile, the projects that I just showed you was going on in parallel. And so I'm going to show you about the convergence of this. We also got a European Union grant, a program and project grant. And this shows some of the same players. There's Anne, there's Nick from the lab, part of my head, Martin again. And this is Anne, this is Nick and Messiah from here and having a tough time in Paris last summer. It's hard to believe. We have a great website if anybody wants to see it. So a little bit about, so our project was on pathogenesis of small vessel disease of the brain. A little bit of background, small vessel disease of the brain contributes to about 30% of the ischemic strokes, which you heard about yesterday, 90% of the spontaneous intracerebral hemorrhages. It's a leading cause of age-related cognitive decline and disability, contributing in a major way to dementia. The risk factors are age, which I don't believe anymore as I'm getting older, hypertension, and the genetic forms of it. Of course, like many of these things, diseases of the brain, they start silently for many years before they manifest clinically. Up to recently, I would say a lot of the mechanisms were poorly understood. No specific treatments, but I'm going to try to show you some of the treatments. And there are a number of genetic causes of small vessel disease. And we initially focused on CADASIL, which I'll tell you about, which is the most common form of small vessel disease. And CADASIL, which stands for cerebral autosomal dominant arteriopathy with subcortical infarcts and leukoencephalopathy. Now I can breathe. CADASIL. It's an autosomal dominant. It's the most prevalent hereditary small vessel disease of the brain. There's about 1 in 50,000, so there are patients here at our hospital. It has all the clinical and MRI manifestations of sporadic small vessel disease. CADASIL is caused by mutations in the extracellular domain of Notch3, which is only expressed in the brain in vascular smooth muscle cells and in pericytes in the brain. And it's also, the mutant is also expressed in blood vessels throughout the body, but the disease only manifests in the brain. And it causes deposits of the extracellular domain in blood vessels in the brain. So a little bit of background. This is a history of a patient with CADASIL. These are years. So their early detection of, depends on, of deposits, when these happen throughout the vasculature, so you don't have to take part of somebody's brain. There's white matter abnormalities early in life, it's 30. Many of the patients, but not all of them, have aura with migraine with aura. And they start having ischemic stroke events early in life. Cognitive decline starts having high cognitive function declines even in the 30s. And this motor disability. And importantly, which is what we study, there's a decrease in perfusion and that's functional hyperemia, which I just described in CADASIL patients and in general in most forms of Alzheimer's and neurodegenerative diseases. So this is our mouse model. It has a human mutation. One of the human mutations that causes CADASIL. The mouse model has the deposits. of the Notch3 extracellular domain. In fact, what Anne has done is she's analyzed the molecular components of the deposit, and from our mouse model and from autopsy samples from human CADASIL patients, and they have exactly the same proteins in the deposit. They have also decreased cerebral blood flow and white matter lesions. This is the mouse model. So it really looks like it recapitulates a lot of the early pathology of the disease. So deficits in functional hyperemia. So functional hyperemia, as I mentioned, is an increase in blood flow to neural activity. So what we commonly do, Mrs. Messiah actually did it, wiggle the whiskers of a mouse with laser Doppler fluorometry, measure the fractional increase in blood flow in the barrel cortex, which is responding to this whisker stimulation. And this shows an example of it. Messiah wiggles the whiskers, blood flow goes up, turn off stimulation, blood flow goes down. Now, if she adds barium ions at 100 micromolar to the cranial surface, these barium is a rapid pore plugger of the inward rectifier potassium channel. So you can see the response goes down by 70% or so. Now, if we go to a CADASIL mouse that's 6 months of age, the increase in blood flow is about half of what that happens in the control animal. And the barium sensitive, inward rectifier sensitive component is essentially gone. And this, we believe, is the capillary mediated retrograde electrical signaling that is gone. And I should also mention, part of our Leduc network and our Horizon 2020 is we also have similar studies done on CADASIL cohorts in Europe and France. And Hugues Chabret, one of our collaborators, and showed that CADASIL patients also have a deficit in functional hyperemia even early in life. And so this simply shows that the increase in blood flow is reduced by about 50%, and it's the barium sensitive, this electrical signaling sensitive component, which is shown in this cartoon. Normally potassium would go up, hyperpolarize, send electrical signal upstream, relax the smooth muscle. And here we use barium as a rapid pore blocker, and it blocks, and I would say in our hands about 70% of functional hyperemia is due to this mechanism. And it's not working in CADASIL. So what's going on? So we tested this in the Fabrice prep by, again, taking an arteriole from within the CADASIL mouse brain with the capillaries attached and spritzing potassium on the capillaries or directly on the arterioles. And what you can see here is that if you put, this is pipette 1, this is the diameter of the arteriole. If you spritz on potassium on the arteriole directly, it's a dilation. If you put it on the spritz it onto the capillaries, response is gone. If you remember the first movie I showed you, where you had this rapid and robust dilation to spritzing on potassium here, completely gone. Consistent with the in vivo data. So we wanted to figure out what the answer is. And I'm not going to show it, but Osama Araz in the lab showed that in CADASIL that the current density of this potassium channel in the capillary endothelial cell is reduced by about 50%. And we knew from the literature that this ion channel, the inward rectifier 2 family members, absolutely requires a phospholipid, phosphatidylinositol 4,5-bisphosphate, to be active. So if called PIP2, if PIP2 goes down, the channel becomes inactive. So we went to test that, and this is a remarkable experiment. This is a CADASIL, this is the arteriole here, the capillary here. Fabrice then picospritzes potassium onto the capillary, no effect in CADASIL, as I just showed you. If you do it on the arteriole, big dilation, which is. Now what he does is he puts PIP2 in the bath for 15 minutes. This is the capillary stimulation, same preparation. The remarkable feature is here is that this deficit in the mechanism that causes an increase in blood flow to neuronal activity that has been going on in the mouse for 6 months, Fabrice has corrected that in 15 minutes by giving the agent that turns on the inward rectifier potassium channel. I should say, I'm not showing it here. Osama also tested it directly on the ion channel currents, and PIP2 restored them to normal. So that's sort of mind-boggling. So the Messiah tried this in vivo with the whisker stimulation shown here and so this whisker stimulation shows the increase in blood flow in a cortical volume. And so it's a whisker stimulation. This is in CADASIL, goes up. It's half the normal level. She then puts PIP2 into the bloodstream, IV, and it's an anion. So the thought is that it wouldn't cross the blood-brain barrier, but would incorporate into the endothelial cell membrane. And she restored the normal response to whisker stimulation in about 20 minutes of application IV of PIP2. And this was all through this inward rectifier mechanism. So just to carry on this theme a little bit longer, Amreen and Osama in the lab decided to look at a different neurodegenerative disease, an Alzheimer's mouse model, and see what happens to blood flow there. And so we decided to use a mouse model, the 5X FAD mouse model, which overexpresses 5 human mutations associated with familial Alzheimer's disease. It shows Alzheimer's disease, amyloid pathology, plaque formation, impaired cerebral blood flow, which is characteristic of the disease. And this shows also the amyloid beta plaques. These mice are 1 year of age. And this shows you could really say, this is what we saw with CADASIL. Normal response to wiggling the whiskers, blood flow goes up in the somatosensory cortex, bam, it goes up. In the Alzheimer's mouse model, it's reduced by about 50%, very reminiscent of the CADASIL situation. So if barium is put on the cortical surface, which blocks this retrograde electrical signaling, it reduces the increase in blood flow by 70%, which is what I showed you before. And what disappears in Alzheimer's mouse model is the barium-sensitive electrical signaling inward rectifier-mediated pathway. And if spritz on potassium onto the capillaries, downstream and arterial, this shows red blood cell flux and the control goes up, all cases. In the Alzheimer's mouse model, no effect of potassium. So it looks like everything the defect is very reminiscent of CADASIL. This shows current recordings. This is a decrease in the current, and Alzheimer's reduced about 50%. This is essentially what we saw in the CADASIL mouse model. Osama puts PIP2, which is absolutely required for the activity of the potassium channel, into the bath, and this is an increase in current. It restores the normal inward rectifier current density within 10 minutes. This is just summarized here. So it's pretty remarkable. Now, unlike the CADASIL, there is a literature of abnormal regulation of phosphatidylinositol 4,5 bisphosphate, PIP2, as well as inositol phosphonostitide metabolism in aging and Alzheimer's. So there's a linkage. I got another prop here. This is lecithin, and lecithin is over the counter, contains a lot of phospholipids, and one of the ones it contains is phosphonostitide. And they bill it. I haven't taken it yet, so I'm afraid to, but they bill it for improving cognitive function. So if I start fading now, I'm going to take one. So does PIP2 correct functional hyperemia in the Alzheimer's mouse model? Again, IV injection. The response of the capillaries and the increase in red blood cell flux to potassium is now restored. And remarkably, here's the functional hyperemia. Reduced in Alzheimer's. With PIP2 in the bloodstream, normal functional hyperemia is restored. You can see that the increase in blood flow is almost more than doubled, shown here. and it's barium sensitive, meaning it's through this mechanism. And pretty remarkable. So impaired cerebral blood flow and capillary endothelial cell inward rectifier channel function is impaired in Alzheimer's and corrected by PIP2. So this sort of summarizes it all. There's a smooth muscle cells, downstream capillaries, the inward rectifier potassium channel 2.1. It's a sensor of potassium. It requires PIP2 for activity. No PIP2, the channel's inactive. So our data suggests the channel density, the channels are there, but just not functioning because of a lack of PIP2. When they're turned on by potassium, it sends electrical signal upstream, vasodilation. So PIP2 is necessary for the capillaries to sense neural activity, to send it to upstream to deliver blood. An Alzheimer's disease mouse model and our small vessel disease CADASIL model, there's impaired cerebral blood flow, reduced functional hyperemia is reduced by about 50%, and this is due to impaired inward rectifier potassium channel activity in the capillary endothelial cells, and this mechanism is turned off. And if we add PIP2 back, we can restore the channel activity, the ex vivo response to potassium, and in vivo response to neural activity. So one little bit of bling from experiments from Tom Longden and Amreen and Grant Hennig's analysis, just as a little teaser. This shows a volume of a cortex, a mouse cortex with its blood vessels that had the dye in it. This shows Grant rotating. It's about 425 by 425 by 50 microns. And now what fades away is now mass all the arterioles, and now the capillaries are left. And we have a genetically encoded calcium biosensor in the endothelial cells. And this now is time, 4 minutes going by, and the capillaries are having these enormous intracellular calcium signals that range over a factor of 10,000-fold, happening all the time in vivo. So that's another project we're looking at. What are these things doing? We think they're involved in local control of blood flow at a more microscopic level. So a little few thoughts. The holy grail, if we restore normal blood flow control in small vessel disease and or Alzheimer's, will that improve cognitive function? I hope so. I also want to throw out the idea that PIP2 is a master regulator of cerebral blood flow within the brain, and that it is compromised in a variety of cerebral vascular disorders. I showed you approaches to rapidly restore normal cerebral blood flow in small vessel disease. I would say this also has implications for restoration of small cerebral blood flow following ischemic stroke, when you want to make sure that the blood is being delivered to the right place at the right time. This is, of course, dates me, but this is a Monty Python, The Search for Holy Grail, John Cleese, and now for something completely different. Well, maybe we're just really barking up the wrong tree. It's not about delivering oxygen and glucose, but actually it's all about removing toxic metabolic waste that the neurons are generating all the time. So that's another sort of another side that we hope to look at in the future. Now I want to acknowledge people, great people in the lab over the years. This shows a meeting we went to a few years ago in Woods Hole. It's Tom Long, who did a lot of the calcium signal work, as well as the capillary work. He's now a faculty member, tenure track at the University of Maryland. Dan Collier was working on something else, on histones and damage and trauma. He's now a faculty member, tenure track at the University of Tennessee. Albert Gonzalez is working on another project. He just left for the University of Nevada, tenure track faculty position. Osama's still here, and he loves lobster. I gave him an extra one that day. I said, I can't pay you much, but you can have more lobster. Then our Leduc network, we go all over the world. This is Fabrice, so the Fabrice fame, drinking red wine in Munich. We had a lab meeting with at Hadrian's Wall in England last February, a year ago February. Osama there. This is our Leduc meeting in Paris, drinking Guinness beer with Fabrice, Gros, and Masayo, and Albert. David Hill Eubanks, longtime collaborator. Susan Amar from the NIH, friend. Adam Greenstein from Manchester. View, there's our lab. This is Anne Jutel. She won the Brain Prize last year, in May 2019. at the, this is in Copenhagen. This is the Crown Prince of Denmark. I forgot what his name was. These are our collaborators. I think his name was Freddie or something. Messiah, Tom and Albert. Lots of funding from the NIH, the European Union, the Duke Foundation, support from the Cardiovascular Research Institute, and more people from the lab. John Lederer again, 25 years later, chilling in the Shelburne Farms with Fernando Santana. Maria's doing great work. Tom Hepner, working on urinary bladder. Collaborators, Jerry Herrera, former graduate student, back 20 years later. He is now as a faculty member in pharmacology, just got his R01. Terry and Dan, technicians. Okay, and thank you, and thank UVM. Thanks, life here has made it possible for this to happen to me. And there's Dave and I celebrating last year. We gave us each other hugs every day until the pandemic hit. And now elbow bumps, I guess. So hopefully we'll go back to giving hugs. But it's really, I mean, because Vermont's such a special place, the colleagues, friends, family, everything has been great here. And let's continue the good work here and our friendship and make things happen. Thank you very much.
Well, thank you very much, Mark, for a very stimulating presentation. I'm a longstanding Monty Python fan, so it was great to see that incorporated in your slides. We've got a host of questions coming in. Are you ready to take some of these on, Mark?
Sure.
First one is a question about retrograde signaling.
Yeah.
In relation to cerebral blood flow. And the question's wondering whether if parasites were removed from the capillaries in your capillary arterial prep experiments, and if not, how would you separate the potential impact of the parasites on retrograde signaling?
The ex vivo prep. So this is back up. Parasites are mural cells that are in the basement membrane that surround all capillaries. They are electrically coupled, recent experiments suggest they are electrically coupled to the endothelial cells. I think in ex vivo prep, I think those parasites are damaged, but the clean tool was genetics. We knocked out the inward rectifier potassium channel only from the endothelial cells, and communication stopped ex vivo and in vivo. But they're clearly playing an important role I think they're playing an important role well, here's our vision. Neurons are active, send electrical signal up, opens up the penetrating arterioles, blood comes down, flows down into the capillary network. But it has to be distributed in all the branches to the active neurons. And there, I think the parasites are playing an important role in saying, okay, it's almost like you're feeding, it's equivalent number of neurons that are being fed by one of these arterioles of about 20,000 neurons. So imagine you have a field of 20,000 plants that have different metabolic needs or different demands for water. You have one pipe coming in, you open up that pipe, but then you have to direct it to the plants that need the water the most. That's what the contractile parasites are doing, I think.
Okay, great. We've got lots of good questions coming in, and almost all of them include compliments on your research and on the presentation. Next question, can you provide an idea about what specifically makes resting membrane potential different among different cell types, like neurons versus endothelial cells?
Yes, the resting membrane potential of endothelial cells is about minus 40 millivolts. A typical neuron is about minus 80, minus 90 millivolts. And smooth muscle cells are also much more depolarized. And there's a variety of, this is set by the activity of the different ion channels and the different cell types. And we made some inroads. Nick and Tom Hepner and Maria in the lab have been able to measure the membrane potential with microelectrodes of both pericytes and capillary endothelial cells. And so for the mechanism to work, the membrane potential has to be, of the endothelial cells, has to be very positive. Because what happens when the potassium channels are turned on, the membrane potential becomes much more negative. And we've measured it directly. It's about 30 millivolts more negative. And so if you remember your neurobiology or Rick, cardiac electrophysiology, the action potentials are a depolarization that is triggered, that can regenerate and can propagate. This is like an inverted action potential. Instead of being depolarizing, it's hyperpolarizing, and by virtue of this ion channel, the inward rectifier, it can regenerate. So,
great. Next question. Says, may have missed it, but do you know if plasma PIP2 levels are affected in CADASIL patients or patients with dementia?
Yeah. Well, that literature I showed briefly has evidence for dysregulation of PIP2 in Alzheimer's. I don't know. I don't think anyone's looked at it in CADASIL patients, although the general, hypoperfusion and decreased functional hyperemia is seen in patients. So, and I think when the people were looking at the phospholipids in Alzheimer's, they weren't looking at the mechanism we're looking at. The PIP2 is, by the way, is a minority interleaflet phospholipid that regulates a number of transporters and ion channels, and also regulates endocytosis. So I think it was originally looked at for other reasons.
Okay. Next question. Is there a connection between PIP2 and regulating eNOS function mediating dilation?
Yes. So that movie I showed of the calcium signals in the capillary endothelial cells is in response to local neural activity that then turns on a protein in the cell membrane, a receptor protein that chews up PIP2 to make another lipid, IP3, which causes calcium release. So every calcium signal you saw there in vivo basically represents a local depletion of PIP2, but that causes calcium release, and calcium activates nitric oxide synthase. So every one of those calcium signals I showed you in that movie represent, starting with the neural activity, releasing a transmitter that turns on the endothelial cell, chews up PIP2, that releases, then leads to an elevation of IP3, which causes calcium elevation. That calcium turns on nitric oxide synthase. So you can also view those those calcium signals I showed you as local points of nitric oxide production that would then relax the overlying mural cells.
Great. Next one's a fun question. It says, if PIP2 is given to a wild-type control mouse, would the mouse show improved cognition, learning, and memory?
Hey, what do you think I'm doing, man? I'm not taking any chances. That's a good question. We have different mouse models, and the thought would be to try to see, of course, there's expense and long-term, what would be the impact of improving cerebral blood flow over, like, say, 6 months. But right now, when we give it to control animals, PIP2 does not increase the whisker-stimulated increase in blood flow is not further increased in a healthy animal by giving PIP2. Now the question is, 6 months is not a really old mouse. So the question is, even a year, if we gave it to a 2-year-old mouse, would it improve cognition where they can definitely show it's decreased?
Okay, next question. With impaired blood flow and increased toxic waste, what else happens? And then the question is, any markers of inflammatory response?
It's a good question. In CADASIL, no. If we look later in life, maybe. I don't know. I mean, this is sort of the riddle, folks. It's sort of the paradox. It appears that the brain usually delivers more oxygen than you need, and that this functional hyperemia is really an oversupply of oxygen. So the question is, well, why? And one would say, well, you don't really want your neurons to live on the edge of metabolic crisis. And the second thing is, obviously the animals are alive with half their functional hyperemia gone. But then the question is, what happens if this goes on for long periods of time? There must be some compromise. So I don't know yet, but lots of interesting things to explore.
Okay, another question's come in. And specifically, what would be the strategy to increase brain blood flow in patients by activating KIR 2.1 channels using PIP2? Can PIP2 be administered pharmacologically or via genetic methods?
Well, we introduced it IV, and it didn't have any. So when the experiments are being done, heart rate's being monitored, blood pressure, and all sorts of other things. But this is not a long-term application. And one of the remarkable features, this gets to the pharmacology, which there are a lot of different, you can get PIP2 in a whole bunch of different varieties. You can get ones that have different longer side chains, more water soluble, less. So there may be ways of delivering it, or you might be able to deliver the precursor of PIP2, that would then increase PIP2 in endothelial cells. So there are a bunch of different approaches that you could in vivo potentially administer PIP2 for long periods of time, or a precursor. So we haven't gotten there. I should say we have patented it. So hopefully it'll make the College of Medicine a ton of money sometime when I'm gone. And you can live in the style you want, Gordon.
Yeah, actually, that was one of my questions, because clearly there's some entrepreneurial activities, opportunities around this. Have you been approached already by potential sponsors?
No, but I haven't done it. Other than the patent through people here,
Right.
but through the Leduc Foundation, they have their own venture capital arm called Broadview Venture Capital, and they're in Boston. So I was going to approach them because their idea is to take Leduc network recipients and try to translate some of these things. It's all cardiovascular and neurovascular too.
Right.
So that was going to be my next step with this.
Yeah, sounds great. Okay, another question. To combine your passions, Mark, do endurance athletes have better blood flow to the brain, and might they benefit by having better cognitive function and lower risk of dementia later in life?
That's from Dave Washer, I bet.
I'm not telling.
No, of course that's true, because, that's why Dave and I go running every day, otherwise we'd be in really bad shape. Well, actually, I'm joking, but there've been a number of physiological studies showing that improved cerebral hemodynamics for people that exercise, and that they basically, somebody that does what we do, they might have cerebral blood flow profile of somebody 15, 20 years younger. Whether that staves off dementia, I hope so. As I said, we have a project in the lab. We have a project in the lab on urinary bladder as well. So I often say, well, we're working towards slowing dementia and urinary incontinence. So here's the question: which is more important for me for my research activity? If I'm demented, I don't know I'm incontinent. But if I'm incontinent and not demented, that's bad.
All right. I think on that note, there's no more questions. So I think we will wrap up. First, Mark, I want to congratulate you on a just awesome presentation. And we'll have everybody give you a virtual hand here. But really appreciated, and carry on the good work. Like I say, most of the questions and comments that have come through, we're really all just congratulating you and the fantastic work that you're doing. So thank you so much. I want to also transition and acknowledge everyone that helped me put this entire program together, and that includes Bruce Kimball, our virtual Zoom guru, Liz Dorman from Communications, Aaron Montgomery, Chris Berger, Vicki Gilway, and many others. It was a massive group undertaking. As I've said before, I do want folks to reach out with comments, suggestions, questions. We will get copies of the State of the College address slides and awards out to everyone. I do want to recognize all the faculty reviewers who assisted in reviewing multiple award nominations. It was a lot of work and greatly appreciated. And lastly, again, this is a very challenging time for everyone. It just seems like everything in the world is changing around us. And again, I want to acknowledge all of your contributions in research and keeping us safe and healthy here in the university community. And I wish you all the best and thank you for attending our festivities and have a good evening. Bye, everybody.
All right, thank you. Thank you.
Take care, Mark.
Thanks, Gordon. Thanks, Rick.